Li from UCLA, talking about scImpute, an accurate and robust imputation method for single-cell RNA-seq data.
It's not showing up.
Oh, let's go through the one in, sorry, there's a folder here. This is yours here. And then it will go over here. And is it Control L, I think? There you go.
Okay, thank you very much. I'm Wei, and I'm currently a fourth-year PhD student at UCLA Statistics. And I'm very happy to be here and introduce scImpute, which is an accurate and robust imputation method for single-cell RNA-seq data. So as some other speakers have demonstrated, the single-cell sequencing technologies have revolutionized biomedical sciences by allowing us to study many biological questions at single-cell resolution. Especially, single-cell RNA-seq allows us to evaluate and compare the transcriptomes of individual cells, and thus enabling the identification of new cell subtypes and marker genes. Single-cell RNA-seq starts with the isolation of individual cells, followed by the library preparation step. Then we can sequence either the full length or only one end of the mRNA molecules, and then the sequenced reads can be mapped back to the reference genome, and we can summarize the read count matrix to represent the single-cell expression profiles. So if we compare single-cell RNA-seq with the traditional bulk RNA-seq, a major difference is that in bulk RNA-seq, what we observe is only one sample containing millions of cells, and the gene expression signals are averaged across cells. But in single-cell RNA-seq, we can actually study cell-to-cell variation in terms of transcriptome dynamics, and this allows us to quantify the intra-population heterogeneity at a much higher resolution. However, this high resolution also comes at a price. If we randomly select two cells in the same population and plot the gene expression profiles in two cells, we should expect to see a very strong correlation. But usually in single-cell data, we will see that many genes that have moderate to high expression in one cell would have exactly zero count in the other cell. And these are so-called dropout events. More formally speaking, a dropout event occurs when a transcript is expressed in a cell but is not detected in its mRNA profile. And these events happen because the amount of mRNA molecules in the individual cells are so limited that some of them are not detected in sequencing. Now, more than 20 protocols have been developed for single-cell RNA-seq, and the frequency of the dropout events depends on different protocols. For example, if we use the Fluidigm C1 platform, then each time we can sequence hundreds of cells, but with a relatively small proportion of dropouts. However, if we switch to the droplets-based protocols such as DropSeq, even though each time we can sequence much more cells, the proportion of dropouts is also much higher. Many statistical methods have been developed to help us analyze single-cell RNA-seq data, but it is still necessary to have a genome-wide explicit imputation method, since many downstream analysis, such as differential gene expression analysis, relies on the accuracy of gene expression measurements. Otherwise, we will have many false positives or false negatives in our discoveries. So this motivates us to develop scImpute to address the excess zeros in single-cell RNA-seq data. And a key consideration in scImpute is that we don't want to impute all the gene expression values because some values are already accurately measured, and imputing these values may introduce new bias into the data and eliminate biologically meaningful variation. So the key idea of scImpute is that for each gene, we want to determine which gene expression values are most likely affected by dropout events, and then for each cell, we only impute the highly likely dropout values by borrowing information from the same gene's expression in its similar cells. For example, if we want to impute the gene expression in cell J, we first divide its genes into two sets, set AJ and set BJ. AJ represents the genes that are highly likely dropouts, and genes at BJ are accurately measured. Then based on set BJ, we find the similar cells of cell J and impute the gene expression of set AJ by borrowing information from these selected cells. The input of scImpute is a normalized and log transformed gene expression matrix, and after imputation, scImpute outputs a gene expression matrix with the same dimensions. To introduce how scImpute works, I'm plotting here the expression distribution of 3 example genes in 3 subpopulations of the human embryonic stem cells. And we can see that first, the distribution of the same gene's expression is different in the 3 subpopulations. And second, all these distributions present the bimodal feature, where the first mode largely represents the dropout component, and the second mode represents the true expression. Therefore, we divide the imputation process into 3 steps. In step 1, we try to detect the cell subpopulations and outlier cells. In step 2, we identify the dropout events. And in step 3, we try to impute these dropout events cell by cell. In step 1, we start with dimension reduction by principal component analysis to reduce the effect of dropout values. And then we calculate a distance matrix between the cells, and those cells that do not have any close neighbors are identified as outlier cells. These outliers can be either a result of technical error, or they may represent the rare cell types, and outliers are removed from the imputation step. Then we perform a spectral clustering on the distance matrix, and then the cells that are in the same cluster as cell J are denoted as its candidate neighbor set, and we will use this information in the imputation step. Then in step 2, to identify the dropout events, we use a mixture model to describe the bimodal feature of gene expression. For gene I in cell population K, we suppose that we use a gamma distribution to model the dropout component, and we use a normal distribution to model the correctly amplified component. We use this parameter lambda to denote the dropout rate of gene I in this cell subpopulation K. Then with probability lambda, the gene comes from the dropout component, and with probability 1 minus lambda, the gene comes from the correctly amplified component. The parameters in this mixture model can be estimated using the EM algorithm. Then based on the estimates, we can calculate Dij, which is the probability that gene I comes from the dropout component in cell J. And this Dij is also defined as the dropout probability of gene I in cell J. So to distinguish the two concepts, each gene I has an estimated overall dropout rate lambda ik, which does not depend on the individual cells. And the estimated dropout probabilities Dij may vary among different cells. So here, the blue histogram represents the observed expression distribution in data, and the black lines represents the fitted expression distribution given by the mixture model. Then with these estimates, we are now able to identify the dropout events. Basically, we set a threshold T on the dropout probabilities. Then the genes with dropout probabilities larger than T are denoted as set AJ, and they're in need of imputation, while the genes with dropout probabilities smaller than T are denoted as set BJ, and they have accurate gene expression with high confidence. By default, T is set to 0.5 in our program, but it can be adjusted by the users based on their desired level of imputation. In the bottom, I'm also plotting the distribution of dropout probabilities in 4 randomly selected cells, and we can see that most values are close to either 0 or 1, so this shows that scImpute is robust to the selection of this threshold. And finally, in the last step, we can now impute the dropout events cell by cell. For example, for cell J, we first select the similar cells of cell J using the non-negative least squares regression, since the candidate neighbor set resulting from the initial clustering may not be very accurate. Then, for gene set BJ, its value is kept as the observed value, while for gene set AJ, we impute their gene expression values using the predicted values in the regression step. And we can do this cell by cell to complete the imputation process. We evaluate the performance of scImpute using multiple data sets from different perspectives. And the first example I would like to talk about involve about 3,000 cells from the mouse cortex region. And this data set contains 57 ERCC spiking transcripts, which are the synthetic transcripts with known concentration. We can see that, so in the first row, it represents the raw data, and we can see that in these 4 randomly selected cells, each cell contains multiple dropout events where the spike ins have 0 read count. However, after we apply scImpute, the read count in single cell data has a better correlation with the true concentration levels of the spike ins. This shows that scImpute is able to recover the dropout events. And in the next example, it involves a synthetic data set where we assume there are 3 cell types, each with 50 cells, and among a total of 20,000 genes, 810 genes are truly differentially expressed. We use this data set to compare scImpute with the other imputation methods, MAGIC and SAVOR. So here, in the first row, I'm displaying the 2 principal components resulted from different data sets. The first column corresponds to the complete data before we introduce the dropout events, and we can see that the 3 cell types can be clearly distinguished. However, in the second column, after we introduce the dropout events, the relationships among these 3 cell types become less clear. And then in the third column, after we apply scImpute, the relationships among the 150 cells are clarified. In contrast, if we use MAGIC, which corresponds to the fourth column, it introduces this structure that does not exist in the complete data. And SAVOR does not have a very clear impact on the raw data in this example. In the second row, I'm displaying the expression profiles of the 800 truly differentially expressed genes. And we can see that in the first column, the imputed data by scImpute leads to a clearer comparison between the upregulated genes in different cell types. And my next example contains 4,500 PBMC cells from the 10x protocol. And I use this example to show that scImpute can help us define cell types in real data. The proportion of 0 expression in the raw data is above 90% in this example. And I'm first showing the first 2 dimensions of t-SNE results after we apply MAGIC, and we can see that the middle clusters cannot be clearly distinguished without using the true color labels. However, if we compare the raw data with the imputed data by scImpute, we can see that even though the different subtypes of T cells are grouped together in the raw data, we are able to distinguish them after we apply scImpute. And we didn't get the results of SAVOR after overnight running, so we exclude it from this example. And my last example contains both bulk and single-cell RNA-seq data profiled at different time points during the differentiation from embryonic stem cells to definitive endoderm cells. And we will see that the imputed read counts reflect more accurate transcriptome dynamics along the time course. So let me take the gene at the bottom left as an example. The black triangles represents the gene expressions in bulk data, and we can see that overall, the expression of this gene first decreases and then increases along the time course. However, if we look at the raw data, this pattern is covered because of the dropout events. But after we apply scImpute, we're able to review this pattern along the time course. And the other 3 genes also present the same message. We also compared the differentially expressed genes between embryonic stem cells and the differentiated endoderm cells, and we found some enriched gene ontology terms that can be only found from DE genes called from the imputed data, but not in the raw data. And we find that these enriched Go terms are highly relevant to the functions of endoderm cells. So finally, as a short summary, we propose a statistical method scImpute to address the dropout events prevalent in single-cell RNA-seq data. scImpute focuses on imputing the missing expression values of only dropout genes and is compatible with most existing pipelines or downstream analysis of single-cell RNA-seq data. So we have included more examples in our paper, and we implemented the method as an R package, which is available at GitHub. And finally, I would like to thank my advisor, Jessica Li at UCLA, for her guidance and input in this project. And I also want to thank all the other collaborators for their grateful suggestions during this project. And with that, I would like to take any questions. Thank you.
I have two questions. First one, would this scale to 1 million cells? Have you tried it?
I didn't try it, but so in my experience, the running time goes approximately linearly with cell numbers. So I never tried with 1 million cells, but I think in that case, it would take a lot more time. Yeah.
And the other question is, have you tried to run this method with multiple different batches of data? Do you think this might improve batch effects or make it worse?
So as I said, we assume that the input is a normalized count matrix. So if there's batch effect in data, then we suggest that we need to normalize the data before we apply scImpute.
Maybe just a quick question. So in your synthetic data, when you do the dropouts, you actually see still some clusters of cells, like visually in your 2 dimensions. Did you try just removing some expression that basically you don't see clusters of cells anymore? Do your method works in that case?
So you're talking about the synthetic data set, right?
Yes.
Yes, we also have examples where we increase the proportion of dropouts, and in that case, scImpute is still able to distinguish the cell types.
So it's a very nice talk, thanks. I think these kinds of things could be really useful. I just have a comment on the terminology that's being used, not just here, but just more generally. I think the use of the phrase dropout is actually pretty confusing, and especially calling it a dropout event, because it's not like a single event that has caused the zero to appear in the data, in my opinion. It's a series of events. There may be several molecules present in the sample, but none of them were sequenced for one reason or another, and so that's why you saw a zero. The same logic occurs for other low counts, so just because you see one count in the data doesn't mean there was one molecule present in the sample. There may have been more. And so this is a kind of a process that's affecting not just the zeros, but all of the data. And calling it a dropout event makes it sound like there's something special that's coming in and just wiping out your data at certain locations kind of arbitrarily. And I think your model reflects correctly that what's actually going on is that there's low expression there. Exactly. And so there's few molecules, and so they're more likely to miss them all. And I think the field would benefit from, I guess, putting aside the terminology dropout entirely and finding another way of talking about this phenomenon.
Thank you.
All right, final quick question. A very nice talk. I just have a quick question. Like in your second example, you're showing it seems that the single scImpute tends to get a more discrete kind of distribution after imputation. Is that true? Or if you look at the second, if you can go back to the second data set, I'm not sure if that's that.
Is this figure what you're talking about?
Yeah, next one, I guess. yeah, like in this case, it seems that after the imputation, you tend to get more discrete cellular population here. Is that like a better result, or because sometimes like we may appreciate more some like continuous cellular distribution here. If you kind of dissect the one big group into into more discrete subgroups, it might introduce some other issues, I guess.
So the results I'm presenting here is based on the first 2 dimensions of t-SNE results. So with t-SNE, it's not really meaningful to compare the distances between clusters. Usually we only use it to identify different cell populations. So if we're looking at the results of principal component analysis, in that case, it may be more meaningful to discuss the distances between cell populations.
Great. All right, let's thank the speaker again.